Hello everyone. Today we will know about the myoglobin and the hemoglobin. So, uh, this is the structure of the myoglobin, and this is the structure of the hemoglobin. This is the myoglobin. This is the hemoglobin. So the myoglobin is found in the muscle tissue, while the hemoglobin it is found in the RBC or the erythrocyte. So the myoglobin and the hemoglobin, they have their ability is to bind to the oxygen. That means the myoglobin it can bind to the oxygen, and the hemoglobin it can also bind to the oxygen. So but the hemoglobin it is found only in the RBC, while in the myoglobin it is found in the muscle tissue. So both the myoglobin and the hemoglobin, they are the globular protein. Globular protein. So first come to the myoglobin. So what is the globular structure? The globular means simply the round shape, and it can soluble to the water molecule. So here let's say this is the myoglobin. So in the outside, in the blood, so obviously they are the hydrophilic. This is the hydrophilic. So now in the inside, the hydrophobic group has to bond. Why? Because the hydrophobic group, they cannot interact with the hydrophilic group. So that's why the hydrophobic group, they are always present inside it, and the outside the hydrophilic group, they are found, because the hydrophilic group, they can form the hydrogen bond with the water molecule. So this is the structure of the myoglobin. That means because it is the globular structure, the hydrophobic group or the hydrophobic amino group, they are found inside the myoglobin, and the hydrophilic amino acid, they are found in the outside of the myoglobin structure. So now look at the structure of the myoglobin. So in the myoglobin, it is divided into 8 helical structure. That means if this structure, if I expand it to the straight line, then it is divided into 8 parts. That means A, B, C, D, E, F, G and the H. So A, B, C, D, E, F, G, H. This 8 helical structure are found in the myoglobin. That means the A, B, C, D, E, F, G, H. Okay. So 8 helical structure are found. So now at the center of the myoglobin, a group is found, specific group. Here this group is found. This is very important group. This structure, the blue color structure, it is called as the Fe+2. Okay. So this blue color structure, they are called as the histidine. The histidine amino acid. At the yellow color disc-like shape, it is called as the porphyrin ring. Porphyrin ring structure. Okay. So this is the porphyrin structure. Porphyrin ring structure. So that means one is called as the pyrrole. What is the pyrrole group? So this is one pyrrole group. This is another pyrrole group. This is another pyrrole group. This is another pyrrole group. So 4 pyrrole groups are present. That's why it is the porphyrin ring. It is also called as the tetrapyrrole tetrapyrrole structure or tetrapyrrole ring or porphyrin ring. So always the AP can bind to the porphyrin ring, and once the AP bind to the porphyrin ring, it is called as the protoporphyrin ring. So this is called as the protoporphyrin ring. That means here the iron group of the AP is bound to the porphyrin group. Okay. So now this AP can bind to the oxygen, but the oxygen can bind to the AP plus 2 only. If it is present at the plus 2, AP plus 2 condition, then only oxygen can bind. But AP can be present in the AP plus 3. But in that case, oxygen cannot bind. So that means the AP plus 2, or it is called as the ferrous. When the ferrous group is found, then oxygen is bound. When the ferric group or the AP plus 3 group is found, then oxygen cannot bound. So now look at the how the oxygen can bind to this protoporphyrin ring. So this is the structure of the protoporphyrin ring. So it has the 6 plane. That means 1 plane, this is the 2 plane, this is the 3rd plane, this is the 4th plane. So this is the 5th plane, and this is the 6th plane. So look at the 3D conformation. Then total 6 plane are found. So now look at the 5th condition. So here a group is found, and this group is called as the histidine. So this is a amino acid found in the 5th plane. So this histidine can directly bind to the AP plus 2 region. So this histidine is directly bind to the histidine group. So the histidine group, here the histidine group is called as the proximal histidine group. So what is the proximal? That means any object which is found near is called as the proximal. Which is found outer, then it is called as the distal. So here the histidine is found near to the porphyrin ring, that is called as the proximal histidine. And this proximal histidine is directly bound to the AP plus 2 region. So this histidine is also called as the His64, or it is called as the this is called as his F8, or it is called as the histidine 93. Why it is called as the histidine 93? That means in the amino acid sequence it is present at the 93rd position. That's why it is called as the histidine 93. Why it is called as the histidine F8? That means it is present at the F region, this region. In the F region it is found the 8th position. The 8th position found in the F region, that's why it is called as the F8 or histidine F8. So always the proximal histidine, it directly bound to the AP plus 2 region. So now at the 6th plane, in the 6th plane only the oxygen is found. The oxygen is directly bound to the AP plus 2 region, but it should also bind to another histidine group. So this is the histidine group, but this is called as the distal histidine group. Why it is called as the distal? Because it is found far from the porphyrin ring. That's why it is called as the histidine or the distal histidine, or it is also called as the his 64, or it is called as the his E7. So that means 64 means it is found at the 63rd position, or it is found at the E region, and in the E region it is found at the 7th region. So this distal histidine, it is not directly bind to the Fe+2, it binds to the oxygen. And what is the role of the distal histidine? It has the role to help the oxygen to bind to the Fe+2 region. Okay. So now come to the next slide, and this is called as the oxygen binding curve of the myoglobin. This is called as the oxygen binding curve. Okay, and the structure is like that. That means here the P, or the partial pressure of the oxygen, and this is the fractional saturation, or the saturation percentage. And this is the structure is called as the rectangular hyperbolic. This structure is called as the rectangular hyperbolic. Okay, why it is called as the rectangular hyperbolic? Look at this condition, this structure. Here the P, here the 0, that means here not oxygen is found, or the not oxygen is bound. But when it is bound to the first, the one oxygen, then it reaches to the this condition. But the myoglobin can bind to the one oxygen only. The maximum level is 1. While the hemoglobin, it can bind to the 4 oxygen. The maximum level is 4 in case of the hemoglobin, and the maximum case is 1 in case of the myoglobin. So because it binds to the one carbon or one oxygen, that means the maximum is already reached. That's why it is now the straight line. That means it cannot go to the upward again, because the maximum is already reached. So that's why the structure is like that, the rectangular hyperbolic structure. So this is the Hill condition, or this is called as the Hill coefficient, or the designated as n, and here the n is 1. Always remember the Hill coefficient of the myoglobin is 1. So 1 means in any, let's say this is the x, this is the y, the xy graph, if there is a straight line, then the Hill coefficient is always 1. So always the Hill coefficient number, the n number for the myoglobin is 1. So next come to the hemoglobin. So this is the structure of the hemoglobin, and the hemoglobin it is found in the all the vertebrates except the Arctic fish. Arctic means low temperature area. In the low temperature area, the Arctic fish, there is no hemoglobin. Why? Because in the Arctic fish there is very high amount of the oxygen. Why high amount of oxygen is present? Because the water in that Arctic fish, or the Arctic region, they are very low temperature. So that means because of the low temperature, the water movement is very, very slow. So because the water movement is very, very slow, it has the water holds very high number of oxygen. oxygen in that case of the low temperature water. So that's why in the Arctic fish they don't need any hemoglobin, because there is very high level of oxygen concentration, and also they have no metabolic rate. That means the metabolic rate of the Arctic fish, they are very low, and more high oxygen concentration is also found. That's why the Arctic fish, they don't need any hemoglobin. Another example is the earthworm. Okay, you will know after some time. So in the, this is the structure of the hemoglobin. So it has always in the previous slide we have already studied that the hemoglobin is called as the multimeric. Why it is called as the multimeric? That means more than one polypeptide chain are found. Okay, sorry, the more than one polypeptide chain are found. So here in the hemoglobin 4 polypeptide chains are found. So here one is called as the alpha 1, this is the alpha 2, this is the beta 1, this is the beta 2. So 1, 2, 3, 4. So 4 types of the polypeptide chains are found. But normally other peptides are also found. For example, this is the structure of the alpha 2 beta 2 structure, or the HBA. This is found in the adult. In 97% of the adult, the HBA group of the hemoglobin, that means 2 are alpha 2 and the 2 are beta, means alpha 1 alpha 2 beta 1 beta 2, means 2 alpha 2 beta, then it is found in the adult condition, that is called as the HBA. Another is called as the HBA2. Here 2 alpha, but here the beta is replaced by delta. So that's why it is called as the alpha 2 delta 2. It is also found in the adult, but here 1.5 to 3% in the adult, they are found in the this type of the hemoglobin. One is the most important, this type of the hemoglobin, this is called as the alpha 2 gamma 2. That means here the gamma is found in case of the beta or the delta. So they are found in the fetus. So they are found in the fetus. So another is the alpha 2 epsilon. That means here the epsilon group is found. So here they are found in the embryonic condition. Okay, so that means in the adult alpha 2 beta 2 alpha 2 delta 2, in the fetus alpha 2 gamma 2, and in the embryonic condition, actually in the embryonic condition all 3 amino acids, sorry, all 3 hemoglobins are found, but maximum case the alpha 2 epsilon 2 are found in the embryonic condition. So now come to the how the oxygen transports in the blood. So this is the same like structure, that means same heme structure or the porphyrin ring structure is found. In the myoglobin, one porphyrin ring structure is found. That's why myoglobin can bind to the one oxygen only. Here 4 groups are found, or the 4 porphyrin ring are found. 1, 2, 3 and the 4. That's why the hemoglobin can bind to the 4 oxygen. Okay, so oxygen can dissolve to or can bind to the hemoglobin only, normally 98.5%, and 1.5% the oxygen dissolved by the blood plasma, and 98.5% hemoglobin they are found in the, or they bound to the RBC. Now look at the structure, how the hemoglobin bind to the hemoglobin. The oxygen bind to the hemoglobin, and it is called as the cooperative method, or the binding of the oxygen to the hemoglobin is called as the cooperative binding. So what is cooperative binding? Let's imagine to near your home a shop is open. So now let's say your home is found. So now you will go to the shop, then you liked it. So now you come and again you will tell your other friends, so that now he will go there. So now he also liked it, so they or he will tell his friends, let's say third one. Now the third one will go again, so he also liked it, so he will go to the fourth one. So then he will go to the shop. So that means there will be a good crowd in that shop, but first one people should go here, then only they can tell people that yeah, it's a very good person, a good shop, or you can go to this. So this is called as the cooperative method. So that means here hemoglobin also, here it is the hemoglobin. So here the first one oxygen is found, then only again another oxygen is found, another oxygen is bound, then another, the fourth one is bound. But the binding of the first oxygen is very difficult, because before binding of the first oxygen, here the carbon dioxide is present, and the carbon dioxide is bound to the hemoglobin. That means the hemoglobin can bind to the oxygen, can bind to the carbon dioxide also. So the CO2, it binds to the hemoglobin and it forms a ionic bond, or the salt bridge. Salt bridge or ionic interaction, or the ionic bond, or the electrostatic bond. So that means the first oxygen has to disrupt this salt bridge or the ionic bond, then only the oxygen can bind to the hemoglobin. But before that, the carbon dioxide, it already bind to the hemoglobin, it forms the salt bridge. So first binding, or the first oxygen, it binds to the hemoglobin and it has to disrupt the salt bridge or the ionic bond. And this method is called as the cooperative method. That means once the first oxygen is bound, then it will easy for the second oxygen to bind. Then it is more easy to bind the third oxygen, and it will very easy for the fourth oxygen to bind to the hemoglobin, and that is called as the cooperative method. That means they are bind to the hemoglobin by a cooperative method. So now the hemoglobin, let's say this is the hemoglobin, it is found in the two states. One, it is called as the tense or taut, or it is called as the T state. Another is called as the relax, or it is called as the R state. So what is the tense of the T state? What is the R state? The tense means tension for the oxygen. That means in that condition the oxygen is not found. That means here the carbon dioxide is found and it forms the deoxyhemoglobin. So one is the oxyhemoglobin, one is the deoxyhemoglobin. Deoxyhemoglobin, that means here once carbon dioxide is already bound to the tense structure. So that means it is very tension for the oxygen. That means first one oxygen has to bind and that oxygen has to disrupt the salt bridge between the carbon dioxide and the tot state of the hemoglobin. So once the first oxygen, it disrupts this bond, then the one carbon is found, then again second oxygen is found, then third oxygen is found, then the fourth oxygen is found, and that is called as the relaxed. That means now it is very relaxed, they don't need any oxygen. That is called as the relaxed state. So this is called as the T state. 1, 2, 3, 4. That is the T state or the tot or the 10 state. That means here no oxygen is bound. But once oxygen is bound, this is called as the R state. Okay. So now this is the oxygen hemoglobin binding curve. This is the second one. So it is the sigmoidal one, or it is the S shape like that. So this is the sigmoid, or it is called as the S shape. But the myoglobin case, in the previous slide we have studied that it was rectangular hyperbolic. In case of the myoglobin. In case of the hemoglobin, the structure is the S shape or the sigmoid one. So this is the Hill coefficient, and here the Hill coefficient of the N is 2.8, while in the myoglobin the Hill coefficient is 1. This is the Hill coefficient is 2.8 for case of the hemoglobin. Okay. So now one is called as the sickle cell anemia. Sickle cell anemia. So in that case, this is the structure of the beta, this beta 1, beta 2, the beta structure of the hemoglobin. So in sickle cell, this is the sickle cell, this is the normal one, this is the normal, and this is the sickle. So here at the 6th position, this is the normal, at the 6th position a valine is found in case of the glutamate. So that's why it becomes the sickle. So always in the sickle cell anemia, the valine at the 6th position of beta chain is replaced by valine by the glutamate. That means in the normal case, at the 6th position, glutamate or the glutamic acid is found, but here it is the valine. Valine replaces glutamate. So that's why normal structure of the RBC is like that. In the sickle cell anemia, the RBC is like that. It is the pointed structure of the sickle cell or sickle shape. So this is the all about the sickle cell anemia. So now come to the factors affecting affinity of the hemoglobin to the oxygen. So that means the hemoglobin, it can bind to the oxygen, and it is the how tightness is found. That means some factors are also determine the tightness of the oxygen with the hemoglobin. One is called as the pH. So pH means in the low pH, that means in the acidic condition. In the acidic condition, always the carbon dioxide bind to the hemoglobin, because the carbon dioxide can bind to the easily to the H plus. So that's why in the low pH or in the acidic condition, always the carbon dioxide binds to the hemoglobin. That means in that case the oxygen has to disrupt this salt bridge. So that means more oxygen concentration is needed. So that's why, let's say this is the partial pressure of the oxygen, or the concentration of the oxygen. So now it shifts to this one. That means more oxygen. More oxygen is needed for the breakdown of the carbon dioxide with the hemoglobin or the carboxyhemoglobin. This is the carboxyhemoglobin or the carbaminohemoglobin. This is the deoxyhemoglobin, or it is called as the carbaminohemoglobin. When it is present, carbon dioxide is bound to the hemoglobin. When it is present, the oxygen is bound to the hemoglobin, then it is called as the oxyhemoglobin. Okay. So once at the low pH or the acidic condition, carbon dioxide or the deoxyhemoglobin or the carbaminohemoglobin is found. So in that case, a much more high concentration of the oxygen is needed. That's why the oxygen is shifted to the right side of the graph. So that means the affinity in that case, the affinity of the oxygen is very, very low. So once the affinity is very, very low, then the graph will shift to the right side. So that means at the low pH, the oxygen binding curve shifts to the right side, and it is called as the Bohr effect, B-O-H-R, Bohr effect. So what is the Bohr effect? That means in the low pH, the oxygen binding curve of the hemoglobin shifts to the right side, and it is called as the Bohr effect. So this is the one factor that determines the tightness of the oxygen. That means more the pH, less binding of the oxygen to the hemoglobin. That means it will shift the graph to the right side. So next one is the BPG. So BPG means 2,3 phosphoglycerate. So this is called as the BPG. So in the BPG, in the RBC, the BPG is found. This is the RBC. In the RBC, the BPG is found. In the RBC also the hemoglobin is found. So now the oxygen can bind to the hemoglobin. The BPG can also bind to the hemoglobin. But here, before binding of the oxygen, the BPG binds to the hemoglobin. So once it binds to the hemoglobin, again it forms the salt bridge. That means again it forms the T state. That means again the oxygen has to disrupt the salt bridge, then only it binds to the hemoglobin. So always more the BPG, less oxygen affinity. The affinity is low. Then obviously it will shift the graph to the right side. So in the high altitude, like in the mountain, if you will go to the mountain, and here the BPG concentration increases. So because the BPG concentration increases, it binds more and more hemoglobin. So because it binds to more and more hemoglobin, that means there is very little scope of the binding of the oxygen to the hemoglobin. So that's why it is said that in the high altitude, when in the mountain trekking, so there is low availability of the oxygen also. That means oxygen concentration is also low, and that oxygen also cannot bind to the hemoglobin. But because before that the BPG is also bind to the hemoglobin. That's why you will get a respiratory problem if we will go to the high altitude or in the mountain. So this is the mechanism that in the mountain the BPG concentration increases. So this is the all about the third one is the temperature. Temperature means more the temperature, the affinity decreases. That means it will shift the direction to the right side. So this is the all about the hemoglobin and the myoglobin. And one more concern that many of guys are watching just but they are not subscribing it. So please subscribe the channel so that I can boost. Thank you.